Thanks very much for the invitation. I'm delighted to be here. It was years ago when I first came across Frame, and there was a similar event, but Dennis Noble was talking. And I came here in the audience and I was fascinated by everything that Frame was doing. So I'm here representing a lot of people, really. My team is called Computational Cardiovascular Science Team. We are based in the Department of Computer Science in Oxford. And we are a purely computational group. So we really do apply computer science, mathematics to problems related to cardiology and pharmacology. But we work in close collaboration with a lot of people who do experiments in industry, in academia, and also we collaborate with regulators, especially the FDA. We're starting to engage with the European Medical Agency as well, and I'll be talking about what we're doing in that respect. So I represent a very multicultural, diverse group of people who collaborate to develop a new idea for therapy testing. So you are all familiar with the different methods that are used for drug development, and this will be the focus of my talk. But actually, a lot of the concepts can be applied to other types of therapies, such as devices. And perhaps in devices, the concepts I'm going to be presented are more developed, and I'll mention why. So we have been very interested in embedding a new idea for therapy development, which is in silico trials. And especially in silico trials that are based on our understanding of human physiology. And through this idea, we are really now looking to replace part of the animal testing that is done in drug development. When I talk about in silico trials, it's about the use of modeling and simulation for drug testing, for drug efficacy and safety testing. So modeling and simulation is used in many areas of engineering. Nobody would develop a plane without actually first simulating the behavior of or the ability of that plane to fly. So in engineering, in other areas like aeronautics or development of cars, for example, it's very normal to use modeling and simulation. It has been established for many, many years. In the medical therapy side of things, we are way behind. And this is now really taking off, sponsored by regulators like the FDA, more on the side of devices than in drugs, but we are also making a lot of progress in the use of modeling and simulation for drug development. One of the key challenges in drug development and in the evaluation of drug safety and efficacy is the population heterogeneity. If we are going to try to test a therapy and predict what's going to be the outcome of this therapy in the broad population, we need to understand population heterogeneity. What makes us different? How that difference is going to affect the safety and efficacy of these therapies. So one of the ways we can assess that is through understanding anatomical variability. Our shapes are different, and what we do in my group is to focus on the heart. We're trying to understand the effect of drugs on the heart. So one way of analyzing the differences in our heart is looking at how different they are in terms of shape, and how that has implications in different clinical biomarkers that are used in cardiology. Sorry, I just did something wrong. So in order to progress towards this idea of virtual testing, we need to create computer models of the heart in our case. And computer models of the heart that include variability, the variability that is seen in patients, in people. One of the ways we can characterize that is through getting non-invasive images, and this is magnetic resonance imaging obtained in the hospital in Oxford, from a variety of patients. And what we can do through these images is to build anatomical models of the torso of the patient and also the heart. These anatomical models include the variability we would see in different patients. So you can see here that the hearts are of different size and also different positions within the torso. And what we are trying to do with these models is to try to understand how these differences may lead to a difference in the clinical biomarkers that are used in cardiology to evaluate these patients. What we do once we get these anatomical models is to plug in models, mathematical models of the electrical activity of the heart. We know how the heart operates from many years of research and experiments, and we have built mathematical equations to describe how the human heart behaves electrically. So we couple the anatomical models with the functional models, and through high-performance computing and very sophisticated technology, we are able to simulate the electrical activity on different anatomical models from the subcellular level to the ECG. So we are really able to obtain very similar electrical activity than in patients. And we can use that for testing of therapies. So in this case, for example, what we were doing is trying to see how the pose, the location of the heart within the torso was affecting the electrocardiogram. And this is very basic science research, trying to understand when a cardiologist looks at some ECGs or some measurements, how much of that variability she or he sees in the patients comes from a normal variability in the shape of the ventricles. So this is very difficult to do with patients alone, but with mathematical models and computational models of this sort, we are able to do it. And once we build enough credibility on those models through comparison with patient data, we are able to use them for drug testing. So this is kind of the vision. Philosophically, I find it interesting also to talk about this in terms of what these models are. These models have a component of representation. So we're trying to build models that represent a particular type of data coming from the patient. But then they become tools that we can use to test therapies. And this is quite an interesting difference, because we build the models based on patient data, but then those models are used as a tool to probe the physiology and to test therapies. A much more difficult source of variability in data, and that we need to consider for understanding population heterogeneity, is functional variability. So rather than just the shape of the ventricles, how the electrophysiology and the physiology of the human heart is different from one person to another. And this is much more difficult to assess because we don't have non-invasive ways of characterizing that functional variability. So in terms of the shape, we have very sophisticated imaging modalities that allow us to image the heart. But we don't know how different the subcellular processes are between different people. And this is much more difficult to investigate. And in fact, also we know that in terms of drug development and drug safety and efficacy testing, one of the main sources of variability is disease and mutation. So there is a lot of research that is talking about how we are different because our genes are different and how that affects drug safety. But in addition to that, we know that sick people, those who have coronary artery disease, are much more prone to develop adverse reactions, for example. So there is quite a lot of research that has been done in trying to understand the differences between different people, how those differences would lead to adverse outcomes or differences in safety from the genetic and the disease point of view. An additional source of population heterogeneity is circadian rhythms. So not only we are different because our genes are different, but in addition, our hearts are oscillating during day and night just because it's day and night. And there are subcellular processes that are making our hearts oscillating. So this complicates even more the picture of drug safety and efficacy testing, because not only we would need to characterize a person at one particular time, but also those oscillations due to circadian rhythms, the food they eat, the hormones, both male and female. So the picture is rather complicated. So if you tell this to a mathematician, they say, we give up. We cannot really characterize this. This is too complicated. But actually, engineers and physicists can deal with this. We just give up and we look at it in a different way. And I'll tell you what we did with this. So just a bit of physiology, so you can follow me a little bit later. So I've spoken about the way we take images of the heart and we build anatomical models from it. And what we do then, from them, what we do then is to construct these anatomical measures of the heart that are discretized, so they are broken into what could be cells, could be a representation of cells. So this is what makes the problem really complicated to solve, because we have a heart with lots of different cells that we need to solve in supercomputers. And in each of these cells, what we have is a mathematical representation of all the subcellular processes that trigger the electrical activity of the heart. For those of you who are familiar with physiology, I'm talking about ionic currents and calcium dynamics. So each of the cells in our heart is composed of a membrane, and in that membrane there are proteins, and those proteins allow the passage of ions, and those are ionic currents. So our computational models have the representation of all these currents, which are the ones that are affected by drugs. So this is the way we construct our computer models. We take experimental data that measure these ionic currents in human cells, and we develop mathematical formulations of these ionic currents and calcium dynamics based on these experimental data. So this is the subcellular level. We construct these mathematical equations, we put them together into a cellular model. And again, this is for those of you who are more mathematical, these are ordinary differential equations, and they describe the passage of ion channels, or ion species like potassium, sodium, or calcium through the membrane. So we now have a cellular model that allows us to simulate the electrical activity of a human cardiac cell. And we put that together with our anatomical models in order to simulate the electrical activity of the heart from ionic current to whole organ. And this is how we integrate the data that come from experiments of many years of human physiology into mathematical equations that we then solve using supercomputers. There are loads of human cardiac cell models that have been developed for human, for dog, for rabbit species. And for human, we already have cardiac cell models for the different chambers of the heart. So we have human cell models for the ventricles, for the atria, for the sinoatrial node. So it's a very well-developed field, and we share them freely. Those models are available. It all started in 1960 when Dennis Noble, as a PhD student, developed the first human cardiac cell model when he was here in London. So it's a very well-developed field, and there has been a lot of research using these human cell models into the effect of mutations, the effect of drugs. So they have been validated using experimental recordings, and it's a very active field of research. One of the things we did in 2013 is actually, we were looking at this variability I was speaking about before. We were getting data from human experimental preparations, and you can see the red traces here are the experimental data. And this is, in black, a simulation of the action potential of a human cell. So the action potential is the electrical activity of a human cell. The black is a simulation, and the red one is the red ones are experimental data. So we looked at this picture, and we thought this was really revealing. So it was really clear that there was quite a lot of variability in those experimental data, which experimentalists actually hated. So the experimentalists were really scared to show the variability in those data, They thought it was an error, it was bad because their p-values wouldn't be trusted. They wanted to suppress and reduce variability as much as possible. But we thought we had to embrace it if we wanted to understand how different people would develop different responses to drugs. So what we did in 2013 and actually was Ollie Britton, who was a PhD student in my group, was to develop a new approach to modeling that rather than using only one model, we developed the concept of populations of models. So rather than having one single cell simulating, we were simulating thousands of potential cells. And the main difference between those cells, they were all human cells, but the main difference was that those processes, subcellular processes, varied ever so slightly between them. So we were able to have an in silico trial consisting of thousands of cells that are not available experimentally to test drugs. In terms of this variability, I had this slide here because what we did is experimentally started to criticize those data, those experimental data, and they were saying all that variability cannot be true. It's all experimental error. We were saying, look around the room and tell me that we are not all different. So why, if we are all different externally, you're expecting everybody to have the same action potential and the same set? It was just completely ridiculous. But what we had to do is to say, okay, you don't trust those data, give me your data. Let me have a look at the variability in your data. And we engaged in collaborations with a range of experimental labs that are very well respected in the world. And we established those collaborations and we wrote the papers with them. And this is the power of having a big group. You can really change the way the community thinks by doing more work quickly. And this is what we did. So with all these people, so it became established that we had to look at variability and we had to take approaches, computational approaches that were able to give us some insight into variability. So in 2013, it was an interesting date because also the FDA launched the CIPA initiative. And the CIPA initiative this was for pro-arrhythmic risk prediction. And what the FDA sponsored the idea, also with industry, of replacing the Thorough study, which is the first in-human clinical study for drug safety evaluation, to replace that by a combination of computer modeling, like the one I'm describing now, and stem cell-derived cardiomyocytes. So they were asking to replace the first study in human by a combination of in silico, computational modeling and simulation, and in vitro testing. So I was in Washington. I was called to Washington to participate in the meetings, and what they did is to announce the initiative on the first day, with a lot of pharma companies all saying this is crazy. And they wanted to incorporate that in 2 years, 2015. And the next day, we had a meeting, closed doors, experts evaluating whether that was actually possible or not. So for me, it was very interesting to see how you actually push for the idea and then you evaluate whether you can do it or not. So that was at the same time that we published this paper. And what these populations of models really allowed us to do was to go for, this is the response of the human cell to the drug, in this case, flecainide, which is used for atrial fibrillation. And this was the concept that was used until then. Two, when we give a drug to people, you can have different types of responses. In most people, it is okay, and you get a normal action potential, a bit different. In some people, it's a complete disaster, and they can actually die suddenly, and this is what we need to watch for. And in some people, they would have a different type of reaction. So computationally, using all the human physiology we knew by then, we had integrated that into a computer model. We had made a population approach that actually is not possible experimentally because you don't have access to human cells. And we were able to predict these different types of responses. And this is what I was talking about. So it coincided, this is the CIPA initiative that coincided with the release of our paper. And interestingly, also the reaction from industry was complete fear. So they just realized they didn't have the expertise to evaluate whether this computer modeling and simulation was going to be any helpful. So what we did is to engage with them in collaborations. And what we did is to to develop a software that is very easy to use. That our software was academic, so we engaged with funding, using funding from Oxford and EPSRC. We developed a software that is very easy to use. Anybody can use it. We had a student, a high school student, who was able to run the software and develop a project in a week. And we spent some time developing this software that what it does is it runs in silico trials in thousands of cells with having as input the drug effect on ion channels from companies. And this is just 2 parameters in most cases. And what is interesting from this perspective is that you would take the information from how the drug affects the different ion channels in the heart, we plug it into the software, and that tells us what is the response of the population to this drug and whether it can lead to adverse outcomes or not, or whether it's a safe drug. And we also did it in big part from support from Wellcome, but also the National Center for the 3 Rs. And we engaged with a variety of companies, and they were really interested in experiencing this new approach, modeling and simulation, and also in running studies to check the credibility of the software and how they were doing in terms of performance, right? So if we have a panel of drugs, how good are these simulations in predicting the drugs that are risky and the drugs that are safe? So that was a lot of work and actually very stimulating. We wrote quite a few papers with these companies. And one of the main ones was by Elisa Passini, who is a senior postdoc in my group. And this paper was showing the results of one of the first studies evaluating the predictive power of our simulations using all the human physiology we knew. And Elisa won the 3 Rs prize in 2017. The previous paper by Oli Britton also won the NC3Rs prize. So what Elisa did was to take a panel of compounds from which we knew the different effects of the compounds on 3 of the main ion channels in the heart at different concentrations. And also, we knew from these compounds, we knew what was the clinical risk of arrhythmia. And we wanted to check whether using a physiologically, mechanistically meaningful biomarker, which is repolarization abnormalities, which is related mechanistically with arrhythmias, whether we could predict whether these drugs were safe or not. And what Elisa found is that the accuracy of the predictions was nearly 90%. It was really big without tweaking anything. This was just purely based on human physiology, and this was higher than the animal testing they were using in companies. And not only that, the predictions were good, but also they gave us insight into which cells or which profiles would be more prone to develop these arrhythmias. And this is important if we want to move away from labels that talk about the general population, and perhaps some of these therapies can be safe for some subpopulations or types of patients and not others. And because our modeling and simulation incorporates human physiology information, we were able to see that actually the cells that were developing these abnormalities were related to pathologies like heart failure or ischemic cardiomyopathy. So they were also meaningful from that point of view. So this is the main difference between modeling and simulation and machine learning, for example. Modeling and simulation not only gives a prediction, but also explains why and what type of profiles can be more dangerous. And this is something we've been working quite a lot with, engaging not only with industry, but also regulators. And and other people in academia in showcasing that our modeling and simulation can be more than an academic exercise, really, and can make a difference in the replacement of animals. So some of these companies are already taking the software and the initiative a step forward, and they are replacing some of the animals. So we were very happy about that impact. Another type of work we've been doing also in order to progress these in silico trials further has been in trying to understand variability in disease. So even people with the same disease can be very different. And we are stuck in terms of therapy development very often because we don't understand this heterogeneity in disease. And we think we can use computer science for this. So, of course, I'm in the computer science department and machine learning is everywhere. So this might not be common to everybody, but every PhD student who is applying now, half of the PhD students who are applying now to Oxford computer science want to do machine learning. And it's like a big trend. So we are embracing it too. I won't go into the details of what hypertrophic cardiomyopathy is, but it's a cardiac disease that kills young people. And it's very complicated because it affects both the anatomy of the heart, but also its function. So it's very difficult to understand. And within the same family, it's inherited, so it runs in families. And within the same family with the same mutation, there are people who die suddenly and people who are absolutely fine. So it's very difficult to understand. And we were very interested in trying to understand this disease and other diseases to see whether we can progress a bit more into the safety and efficacy of drugs, in particular subpopulations of patients that have diseases like this one. So we engaged with clinicians in the John Radcliffe Hospital that had clinical data that, in families with this disease. They had ECGs, they had MRI, which is the imaging technology I showed you before. And we were trying to understand whether from the clinical measurements like the electrocardiogram, we were able to differentiate patients that had slightly different phenotypes. And again, women leading the work in my group, we have no problem with diversity and gender balance, even though we are in computer science. And what they did was to use machine learning to try to identify these clusters of patients. So within these 86 patients that had the same disease, can we differentiate using algorithms, using machine learning, whether or not we have different types of patients. I won't go into the technicalities. We can talk about it if anybody is interested. But the thing is, we were able to identify 4 different subgroups of patients using machine learning, using an algorithm that selects the features that make them different. And using the clinical information we had, we were able to look at how meaningful those groups were. And we were able to see that some of these patients have abnormal ECGs, but actually don't have a very high risk. And others that have an abnormal ECG have a high risk and differences in hypertrophy. So this machine learning was an example, applied to this clinical data, was an example of how we could separate these patients into 4 groups that were clinically relevant. But we had no clue why these ECGs came from those hearts. We had no idea. And we used modeling and simulation for it. So what we did is to take the images, construct the models in the same way I showed you before, to create anatomical models representatives of each of the groups. And in this case, for example, the patients had a thicker wall, both at the apex, at the bottom, and in the septum, whereas in other cases, it was only in the septum, in the mid-wall. We used our computer modeling and simulation technology to simulate the electrical activity on those anatomical models. Using all the information we have on human physiology, not only under normal conditions, but also in hypertrophic cardiomyopathy. So we are integrating a lot of knowledge into those models. And we tested several hypotheses about what could explain those ECG differences. And we explained them. I won't go into the details, but we were able to recover these phenotypes using modeling and simulation, which meant we were creating virtual copies of those patients that we could use for testing therapy. So we were obtaining, using all the information we had on these models and our simulation technology, ECGs that were very, very much like the ones that were obtained in those patients. And we were able to explain why the ECGs were very different than healthy subjects. So what we want to do with this, and we've done at single cell level, is to create more of these models of different disease conditions and help create populations of models that go from single cell or from ionic level to the whole organ, like in this case, and that have the credibility we need to talk to clinicians about it and also use them for drug testing, and then try therapies on specific disease conditions. So I put here the example of hypertrophic cardiomyopathy, but we have also produced computer models of a heart attack. And we validate those models from single cell ionic level to the whole organ and the ECG to create populations of models that we can use for drug testing. So that's kind of the vision. So what we've done at the single cell level and at the population of cell level to do it at the whole organ level and really reaching towards clinical outcomes like this one. And we combine machine learning and multi-scale modeling and simulation because machine learning can help with analysis of data and teasing out the heterogeneity in the population using clinical data. And multi-scale modeling and simulation can really explain the reasons for that heterogeneity and provides confidence for the findings as well. What we are doing at the moment is going beyond electrophysiology, and we have now models of the heart that allow for testing of contraction. So what we are doing is collaborating now with the pharma companies and our collaborators in looking at contractility, which is one of the big problems in drug testing as well. So we're going beyond the electrophysiology to looking at contraction of the heart. And this is technically very complicated, but it can be really useful if we are really looking at replacement of animal models beyond what we have achieved, because the in vivo models are used for very different endpoints, and one of them can be contractility. So just to summarize, I hope I have illustrated that computer modeling and simulation for in silico trials can be a very powerful and advanced technology. In academia, we have made enormous progress in the last years, and it's only in the last 4 or 5 that I've seen really industry opening up for the replacement of animals. We reached very high accuracy in predicting and explaining drug-induced arrhythmias, and again, this was purely based on our knowledge of human physiology and mechanistic insights into proarrhythmic risk. We didn't tweak the models, we didn't do anything. It was just, we have reached a level of understanding that allows for this to happen. In fact, this is very different to stem cell-derived cardiomyocytes, and it's been very interesting for me also to see the difference in attitude between computer modeling and simulation and in vitro testing. Whereas when our computer models and simulations were failing in the predictions, people were saying the models are rubbish. When the stem cell-derived cardiomyocytes were failing in the predictions, people were saying, oh, we need more money to make them mature. So the difference has been really, really interesting to me. We are much cheaper, really. So, that's why we have engaged with companies in trying to experience the technology by themselves, and now they are opening up to hire people to do this type of thing. Our main strength is always that we focus on human, and we do have a lot of information on human. The fact that we are able to simulate the electrical activity of the human cells and find that we have really credible results but not only at the cell level, but at the ECG level, means that we know a lot more about the human heart that we really are using for drug testing. And our strength is also we can focus on disease. A lot of the adverse outcomes happen in sick people, and in pharma companies are not using disease models. With our models, the way we are developing them to understand disease conditions and integrate the data, we will have a very powerful technology that we can really exploit for this. And of course, understanding heterogeneity, which is key for any medical therapy development, requires diversity of people, expertise, and approaches. And we are really, really motivated to contribute to the replacement of animals in research and therapy testing. So not only through our work and dissemination like tonight, but also engaging with companies through the support of the NC3Rs. And also I'm involved with regulators as well and trying to explain and write guidance document on how to implement these new technologies. And with this, just thanking our funders and of course the people who've done the work. Thank you very much.
Where they are, so I'm just going to speak very loudly. Thank you so much.
I can take this one.
No, you won't. Thank you so much, Blanca. At Frame, we quite often talk about how human relevant methods will ultimately lead to better outcomes for human health than some of our current animal models. And I think that just showed that really, really well and the role that models play in that. So does anyone have
I'll shout. That was great. Fantastic talk. You talked a lot about the biovariability of anatomy, of the response factor as well. Have you started to think about and integrate the variability in drug exposure? Again, based on that population. So my area is pharmacokinetics and population kinetics, PBPK. So that's the next logical step for me for drugs going forward. So have you started to look at that?
Interestingly, yes, but not directly. So we are very proud that it's the companies we have been collaborated with that are taking that forward. So yes, so some of our collaborators in Merck have been looking at combining our modeling and simulation, integrating it with other types of modeling that are much more established. So and it's not us so we wanted to enable the work to be taken forward by others, not only us. So they are doing that. So if you're interested, I can share contacts. Yeah, that's
good. Thank you for the talk. Is there a class of drugs or a particular condition that you tried to model and it didn't quite, you couldn't quite model it? And if it has happened, why, you think? And I've got another one, but I don't know, I can ask, should I ask directly now? Is there, so for example, the human heart, you have a lot of data to build your models on. If you were to think or try to model whether the same drug has side effects in other organs, would you have enough data and computational ability to then try to model the side effects? And could all this new data on single cell analysis from different tissues of humans give you actually data to work on?
So regarding the first question, the answer is yes. So we do have some drugs that we cannot predict the right outcome. And interestingly, it's not necessarily because our human physiology is not known, but it's because of the other aspect that we were talking about. And so if the input data, so if we don't know if the effect of the drug on the ion channels in the assays that are used to generate the input data don't show an effect, we won't see anything. So there's been a shift in terms of what we think is not working now in the last 4 years. So initially, everybody thought the models were the problem. Now, what we think is that the input data are the problem. So there is quite a lot of work that needs to be done in quantifying the effect of the drugs on the different ion channels. And there's been a lot of work done also in terms of standardizing those assays, because there is quite a lot of variability in those input data, especially for the calcium current, which is critical for electrophysiology, but also contractility. So I'm always very cautious, but I am very confident in our understanding of the human physiology, electrophysiology, and how the models are constructed. In fact, we are now releasing another model that is much better. And the problem is in the input data, actually. And if the drug is not doing anything in the ion channel because there's another mechanism, then we won't see anything in the models. And the second question was? Yeah, that's interesting. So there's been work that has been done in liver that I'm aware of. I think in terms of cardiac electrophysiology, we are way ahead. And that's why also regulators took this as an example where we could really move forward in the use of modeling and simulation. So there are other people who are doing the same type of modeling in other organs, probably not as advanced, I would say, but people looking at liver and lungs, kidney. I'm part of a European project, TransQST, that is in part trying to address that. But I would say that we are the most advanced, probably historically. Denis Noble chose the heart. Yeah.
Thanks very much for this talk. Do you see these models being used in the clinic to give personalised medicines at some stage? And if so, how far off is that?
Yeah, well, I hope so. I do hope so. That's a tricky question. So the work we are doing in hypertrophic cardiomyopathy, for example, we do hope it will end up addressing some of the problems there is in patient management. We are starting to work more with the clinicians, and there are other people who are working much more in clinical applications of these models, more related to ablation, for example. So if people have an arrhythmia, they burn the heart, and it's difficult to know where the burning needs to happen. So there are some examples of studies using modeling and simulation that are using already, that are applied to the clinic. Another one is devices. So it's the implantation, for example, of electrical devices in the heart and where to put the leads if you want to have a pacemaker or defibrillator implanted. So there are some examples already of that happening. And as the computer power also helps us, it's easier to generate more examples. So we were a bit stuck because the models are very big, you need supercomputers. But as we have GPUs and other computational platforms, then it becomes easier. So I think this is going to happen. It's happening in some examples. It's going to happen much more, I think. I think the credibility of the models is increasing and people are embracing this much more, I would say.
Very nice talk. I'm interested in your perspective on taking what you've done in terms of taking the complex model and the n equals 62 into a tool that industry can use, which you said a high school student could work with, and then the use for regulatory decision making in dossiers of information coming out of that tool, and whether you've got any insights, because we tend to get the feedback that, oh, it's too complex. And what you've done is try to simplify, but then the criticism sometimes to the regulators is, and then it becomes a bit too much of a black box and we don't understand all of it. And whether or not having the FDA as a collaborator up front actually is going to help that path.
Yeah, so I'm being recorded, right? So I'm...
Yeah, we just need to be careful. We can take it offline.
No, I'm happy. So the collaboration with industry has been very dynamic, and I think we decided to produce this software because it was helping our business as academics who wanted to have an impact. And we We gave it for free to the collaborators. They gave us data. We run lots of credibility studies, and that has raised the profile of our technology. In terms of the interactions with the FDA, initially we had this 2013 release of the CEPA initiative, and we had a meeting with the experts, and then it all went a bit quiet. And they kind of wanted to work on their own solution, protecting that solution. So there was a bit of a disconnection between what the FDA was doing and what the academic community was doing. And the academic community was engaging with industry, and there were different groups. We weren't the only ones working on this, so other people released different solutions, and we were all showing that it actually works, regardless of the solution. So the FDA released their own, so they took my student, and they and she went to work with them, and they kind of developed their solution. They wanted to implement this in 2015, and we still are not seeing that implemented. But what certainly the FDA sponsoring this initiative did is that industry became really interested. So a change has already occurred. In terms of FDA approval, or what is the ultimate solution that would be chosen, or I still don't know. But because they sponsored it, industry took it seriously. There was a lot of controversy. We have progressed quite a lot in 4 years. I don't know what's going to happen with the different solutions. We haven't tried to make our software the solution that the FDA is going to adopt. We're not doing that. But we have already changed the way the companies are thinking about it. And we can talk some more. The dirty secrets.
I noticed on one of the slides that the red, which I think are the simulated ones, were peaking earlier than the. Can you work backwards from the data, from your curves, to say why that's happening and tweak the model?
I love this question.
Okay.
So we saw that discrepancy. It's not the only one. So what also happened is, because we were testing and pushing our models much more with data, we saw that there were a few discrepancies. But the fact that there is a difference at the top was not just a silly feature. So what we found is that when we were simulating the effect of sodium block, contractility was going up. So the calcium transient was going up. And that's exactly the opposite that happens in reality. So if you take a sodium blocker, or you have a disease like ischemia or heart failure that affects the sodium current, your heart is weaker. And that was related to that difference at the top between the experimental data and the simulations. So what we did is to work really hard in producing a new model that addresses that. And we knew there was a big problem, but rather than announcing that there was a big problem with the model to the FDA industry, I said to my group, we need to sort it out. So we're going to say there is a problem at the same time as we give a solution. And that's what we've done. So that's why we have created a new model based on the previous one, and we have shown that the predictions are equally valid. But we have changed that.
And you also, I think on the same thing, with one quite big outlier in blue.
Yeah.
Can you work out why that is?
Yeah, that's easy. That's easy, because usually it's a weaker current, a weaker potassium current. So we can do that sort of thing, is easier. But this discrepancy at the top was actually quite crucial for some of the functionality. But we've sorted it, and our paper is now.
That's good, because it means you've found out more information about them.
Yeah. And but we had to sort it out. We couldn't simply say this is not working, which it was working, but anyway. So, well spotted though. Good eyes.
Could I just ask, if, let's say somebody is developing a new drug and your modelling shows a risk in a minor phenotype, isn't there a risk that the regulators and the companies either wouldn't allow or wouldn't take the risk of developing that compound, which might have a big benefit in the majority of other phenotypes? I mean, it's a matter of balancing risk, isn't it?
But that's exactly what we're hoping for. So what we're hoping for is that we move on from it's affecting these channels, there is a risk for a very small amount of the population, we're going to ban the therapy, when it could actually be beneficial for the rest of the humanity. So that's exactly the type of insights that our population of models can provide. And interestingly, we in that sense, we were using the population and we were getting very weird phenotypes with a very small part of the models. And we saw that those cells had a very low current, IK1. And we thought, oh, that's not possible. They cannot have that such a low current. It's an artifact of the models. But actually, we searched the literature, and there is a mutation that can lead to families having this very low current, and those families are at high risk of arrhythmia. So we do think that there is a lot of information that is clinically valid, and perhaps at some point with that sort of information, with disease models, we can move on from banning therapies for everyone when in fact they could be useful for some people.
So you could foresee licensing for particular phenotypes of patients?
I mean, I don't have that power. I wish, I think we can do that if we work hard and people open up to this idea. I think we could do that. And that's very difficult with animals. Impossible. So I have hope.
But one of the things sort of interested me is if you want to get sort of regulatory acceptance, you always have to go through a soup of qualification or validation.
Yeah.
Horrible words, I know.
Yeah.
But that's the sort of point. People will sort of say, if you claim your models are predictive,
Yeah.
have you looked at the effect of, say, a drug? You predict it, and then you do it against a real life patient to sort of validate it, if you like, your model against a real life situation. Because I think if you don't, people will always look at this a little bit. Like there's two aspects to this. One is the pharmacology, where you're looking at the efficacy of a cardiovascular drug. The second is the safety testing, like they do in the safety pharmacology. So if you're ambitious enough to want to replace all the animal studies, people are going to have confidence that what you do actually really reflects the real life. Yeah. And that seems to me there's a lot of work.
But that's what we did. So what we did is we took actually we've done it several times because the different companies wanted to have it done with their data. So we've done it with Janssen, we've done it with Amgen, we've done it with Merck, and UCB is actually doing it as well. So what we've done is the simulations are predicting abnormalities, and we have shown that those are related to clinical risk of arrhythmia. So we've taken a database for it that has all the reports of clinical outcomes. We've done it for a set of compounds that have this information. And what remains to be seen, and actually is a challenge, is how this technology is incorporated. When you don't have that information, the drugs have not been released to the market yet. So if you do it in drug development, how do you do that? So that's the challenge. What you're saying, we've done it. We haven't gone through qualification, and I have a regulatory qualification for the software, in part because we haven't had time. But a lot of people are saying we should do it, and we should team up with a company to do it. But we've done a lot of the credibility with the different companies. We've found the same amount of information and the same type of accuracy with different sets. So I think there is a lot of hope. Yeah.
It was a combination of your answers to the last two, actually. I just wondered how predictive this was of future heart disease in seemingly normal people at the moment, whether that was going to be an indication.
Well, so future heart. So what we've done is in terms of pro-arrhythmic risk. In terms of future heart disease, one of the areas that have become more interesting is cancer treatment. And that is a particular area where the future heart disease is important to be predicted. People who are taking chemotherapy and develop heart failure in the future. So also the area of cancer treatment is quite important because pharma companies are prepared to take risks with this therapy in terms of arrhythmias and other things. So we're looking into it. It's not easy to predict future risk and chronic effects. We're looking into using transcriptomics for it. I had a PhD student looking at how changes in gene expression in culture cells with therapies were able to predict different types of cardiotoxicity. This is very new to me, but we found good predictions. Again, this is machine learning. So we're using modeling and simulation to try to explain also whether there is a mechanistic relationship. But yeah, the area of cardio-oncology is one in which this prediction of future heart problems is quite important. I'm very interested, and it's a lot of hospitals are establishing that type of practice. It's very complicated.
Unlike the work you've done.
For different reasons, I would say. I think the physiology is less known. The biology is less known.
You also said you got lots of support from the FDA. Then you talked about regulators later on. Have you had any interest from the Japanese regulators or the European the Chinese? Or?
So personally, I haven't had a lot of support from FDA, but the FDA was sponsoring this initiative, and that was very good. The Japanese regulators are around. I haven't interacted with them. I think they like working with Japanese groups, and Japan has strong groups working in this area. I'm sure they are talking.
Any more questions?
Is it relatively, complicated stuff? Is it relatively easy to incorporate different disease models into what you've already established? So then you gain by using previous.
So that's one of the ideas. So rather than, so we can go really complicated in terms of the type of models and supercomputing and using whole organ models, or we can also have different types of cell models that correspond to different disease conditions. And we've done that in the paper on the new model as well. So having a cell model that has ischemia, infarction, heart failure, all the main clinical conditions, mutations. So we haven't done the incorporation in the tool, but we've done all the groundwork that is needed to do that. So we do have very good models. That's what I think our academic work is much more advanced than the uptake it's had in this area. So I think that's one of the directions. So if we have a library of models of different disease conditions and we run simulations for the different disease conditions, can we progress that to having labels that for the different medicines that say, if you have this condition, be careful or don't take it. That's kind of the vision, right? But we can take it forward.
Can I follow that up then? There's quite a lot of research in different models of cardiac disease and health out there in the academic environment. How are you managing to get information to them that actually there are non-animal alternatives that they could be collaborating with you to try and advance their work?
I mean, that's the idea of this NC3R's Infrastructure for Impact Award, where we have a website, we're having a repository of models, we have a newsletter, we have meetings where we showcase this. We also have models now of stem cell-derived cardiomyocytes to combine with experiments. So we are kind of disseminating all of this. And the attitude has changed quite a bit. I remember years ago where I was engaging with the companies, and their attitude was very much, convince me that this is something I want to do. And at some point I said, look, I don't have a problem. You guys have the problem. So if you want my help, we need to sit down and decide how this is going to be done. So now they really engage in a different way, but the power structure was different at the time. And now there is interest, and we through dissemination, you really see uptake as well. That's what we can do from academia.